So good afternoon, and thank you all for coming out to support some science and student research. As Braun mentioned, my name is Natasha Hongsermeier, and I'm an undergraduate student at Morningside College in Sioux City, Iowa. So this is an interesting change of scenery for me here. Today I'm going to talk to you about the role of cilia during early heart regeneration in zebrafish. And this was a project I worked on this summer in the collaborative labs of Dr. Bob Morris and Dr. Vut Yin. So why are we interested in studying this question? Well, cardiovascular disease is the leading cause of death worldwide. To zoom into the United States, 1 in every 4 deaths here is caused by heart disease. To put that in perspective, someone in our country dies about every 90 seconds from poor heart health. Human hearts demonstrate poor regenerative capacity following events such as heart attacks, and the heart often ends up scarring instead of re-healing, and that leads to problems with how it can beat later on. So it affects the ability of the heart to beat well after injury. Zebrafish, on the other hand, are champion regenerators. So you cut it off, they grow it back. They're pretty amazing. So we wanted to look at zebrafish because we know that they can regenerate their hearts fully within about 30 days. So here on this diagram of the zebrafish heart, you can see the ventricle over here. Oops. The ventricle here, and we cut off a little piece of the ventricle here at the ventricular apex called the injury site, and I'll be referring to this as the injury site throughout the remainder of my talk. Also on this diagram of the zebrafish heart, we can see the atrium and the outflow tract. So unlike mammalian hearts, which have poor regenerative capabilities, zebrafish hearts can completely regenerate their cardiac tissue. So if you want to study how something works, you want to study it in an organism that does it best. That's why we chose zebrafish as our model organism. Now, how are cilia involved in this process? Cilia are flexible cellular appendages that come in 2 varieties. You may be more familiar with motile cilia, which move fluid in places such as the lungs, but there's also primary cilia, or immotile cilia, and they stand still and act as cellular antennae to transduce important signals for growth and development. We wanted to understand how cilia might be involved in heart regeneration. So here we have a diagram of cilium. And here's a primary cilium with its axoneme labeled in red here and its centrosome labeled in green on a cell with the nucleus labeled in blue. And primary cilia have recently been shown to be involved in important cellular signaling in embryonic heart development in zebrafish. So since we know that they were involved in growth, we were wondering if they might be involved in regrowth. So that's why we started studying cilia in heart regeneration. Also, recent data from the Yin and Morris lab showed the upregulation of cilia in the injury site at 7 days post-amputation, or DPA. So that leads us to our research questions. The focus on cilia as potentially being important to signaling in heart regeneration led us to ask how cilia might be involved in this process. So here we knew that cilia were upregulated at 7 DPA, and we knew that the heart was fully regenerated by about 30 DPA. And here you can see in this yellow region that, so here's a 0 DPA heart with the injury site here, where we nip off a little piece of the ventricle and it goes back. Here is the yellow clot that forms on a 7 by the time the heart has started regenerating, and this clot is still present and hasn't been replaced with new tissue yet. And by the time we get to 30 DPA, the clot has been completely replaced with new tissue and is innervated with nerves and blood vessels. However, no one had really looked at the 3 DPA time point, which we thought might be a critical time point in the regeneration process. So that was our major question this summer. We wanted to see what was going on with cilia at 3 DPA, and we wanted to look at how the number of cilia and their lengths changed throughout this process. So to explain to you what we learned, we need to do a little bit of reorienting ourselves with the anatomy of the zebrafish heart. So this was the diagram I showed you earlier, with the zebrafish ventricle here, resection site or injury site, atrium and outflow tract. Here's an image of a whole heart that we took this summer. So you can note, as you can see, it's been rotated about 90 degrees from the original diagram. And this is the orientation of the heart that we'll be using for the rest of the presentation. You can see the injury site is down here, the atrium on top, and the outflow tract over here. And whenever you see this little icon in the upper right-hand corner of the slide, that's how I'm going to help us keep track of what part of the heart we're talking about on a particular slide. So in order to investigate our research question, we use immunofluorescent microscopy. Here we have an image of a 3 DPA heart This is akin to this little cartoon over here. So here's the end of the ventricle on both images, and then the clot right here. And here is how we analyzed our images. We used the software program Fiji, and you can see we used different cell markers within the program to show us what structures we were looking for and mark them. So here's the boundary of the injury site. It would be about there. And this is the same image, but it's just been quantified in Fiji. And here are different markers we used to mark cilia and nuclei in this region. So what we were actually looking for when we were quantifying our images is this stain. This is our three-color labeling pattern that we define as a cilium. You can see the axoneme of the cilium labels with acetylated tubulin, and it glows red. The centrosome, which is where the cilium originates, is labeled with gamma tubulin, and it glows green. And the nucleus is labeled with DAPI, and it glows blue. So to remind you of what part of the heart we're looking at again and how we gathered our data, this is what we found. So you can tell in the injury site here, there wasn't a huge change in the abundance of cilia from 0 to 7 dPa. It went up slightly and it went down slightly, as you can see with the y-axis here and the percentage of cells that are ciliated. However, in the injury site, there was a significant increase in the abundance of cilia from 0 to 7 dPa, and it increased in a really nice linear fashion. But what about their lengths? So when we looked at the lengths of these cilia in these locations, we saw that both of them increased significantly at 3 dPa. And they both have similar values between 7 and 0 dPa. So what can we conclude from this? Well, we set out to study 3 dPa hearts. And we noticed that 3 dPa might be a critical time point for when cilia lengthen. And in the outflow tract, we noticed that cilia at 0, 3, and 7 dPa increase, or they remain about steady in abundance, but their lengths increase and then decrease. When we looked at the injury site, we saw that at these same time points, the cilia abundance increased, and their lengths also increased and then decreased, just as they do in the outflow tract. For future directions, we would like to support the data that we see with our immunofluorescence with molecular biology using quantitative PCR. We have some preliminary data, but we'd like to build upon the data set that we have. We'd like to repeat our immunostaining and quantify additional images to bolster our data set. Eventually, we would like to knock down cilia and see how that affects the regeneration process. And finally, we would like to confirm preliminary observations of cilia in the blood. So this is something that was really interesting. We did not expect to find. I want to show you real quick our preliminary data on that. This is a 3 dPa heart, to remind you. And this is what we found in that 3 dPa heart. So in the clot, we found about a 5-fold increase of cilia in the blood clot compared to just circulating blood. And this is really interesting because in humans at least, red blood cells are the type of cell that we do not expect to see cilia on. So maybe that's not true in zebrafish. We're not really sure yet. We're not sure of the significance of what we're seeing here. But it was a fascinating discovery, and we really look forward to pursuing that in the future. So to summarize, while the mechanism of heart regeneration in zebrafish continues to be elucidated, we do believe that cilia appear to be involved, and they might even play an important role in this process. With that, I would like to acknowledge the people who made this work possible, including my research mentor, Dr. Bob Morris, Nina Badger, our brilliant and helpful high school student in the lab, Walker and Hans, who were previous students of Dr. Bob's and upon whose research we were building, Dr. Vut Yin and the Yin Lab, especially Mary Lynn for helping us design our primers, and to Ashley Smith for helping us run qPCR, and of course my funding source, the NSF REU grant. And with that, I appreciate you coming to my talk, and I will take any questions you may have. Sorry, yeah.
So do you have any genes that you're thinking about that might relate to the lengthening of cilia? And how might those relate to during development? Cilia may also be growing, are they analogous to ours?
So the question is, do we have any genes that might relate to cilia length, and how we can examine that first off? And we chose 4 genes that we know are involved in ciliary, we think are involved in ciliary expression, and that we would examine with qPCR. If I had to hypothesize which of those are involved in lengthening, I would think that the structural genes would be good ones to look at. So we chose IFT88, which is important for the microtubule transport region of the cilium, and also another structural gene for kinesin that basically walks on the microtubules. So if we can figure out, I would think that if those levels are upregulated, even in comparison to where there are cilia, but maybe not as long, we would see greater levels of expression for those genes.
I have, if I may, two questions. You showed very nicely this increase in cilia. Which cell types of the heart, myocardial cells and cilia, whose muscle cells, do you think these cilia are on? And my second question would be, are there white blood cells, monocytes infiltrating in these wounds?
Both very good questions. So I'll answer your last question first. So you talked about the possibility of maybe white blood cells infiltrating in the injury site that we're studying. That's certainly a possibility. We do not know which type. We think that they're red blood cells that we're looking at in the clot that we found those cilia on, but we haven't done any analysis to determine for sure their identity as a blood cell. And also the question about which population of cells is throwing out these cilia. We have some preliminary qPCR data on that. We've done some qPCR ones with FACS-sorted cells, so cells that are divided into different populations based on their genetic markers. We did that for endocardium, epicardium, and cardiomyocytes. And the data we got data, but it wasn't consistent across the board. We couldn't make as much sense of it as we'd like. So for our future directions, that's another thing we'd like to optimize, is figuring out which population of those heart cells is actually producing those cilia. If we had to hypothesize for now, we think it's the endocardium, because it looks similar to the stain when cells that are labeled as endocardium cells show up on the outer edge of the heart. That looks similar to where we're seeing cilia.
Is the injury to the heart, how was that created? Is that analogous to an ischemic injury you see in the human heart?
More of an ischemic injury, we don't do this, but you can produce more of like simulating a heart attack by cryo injury, so damaging the heart by freezing it, touching it with a little point of freeze. And what we do is resect it, so we nip off a little piece of the ventricle and actually remove about 20% of the ventricle, and it regenerates. Mm-hmm.
So I was wondering if we looked at microRNA expression in this regenerative response. And the reason I ask is because a few years ago I remember reading a paper about using near-133, which is downregulated, and it seems to increase the regenerative capability. So I don't know if you've seen anything like that.
That's a great question. So the question is about, have we looked at microRNAs in this process? And one of the labs here is looking, the Yin lab does look at a lot of microRNAs, but we haven't specifically in this project. One of the reasons we did choose the 3-DPA time point to look at is we know there are a lot of really important microRNA, there's a lot of important microRNA activity going on at that time. So that's one of the reasons we chose it to look at with our immunofluorescence, but this project specifically did not look at that, no.
Still have a little bit of time, so I'll ask one last question, perhaps. I'm intrigued by this erythrocyte or red blood cell presence of cilia. And so there's an investigator on the back wall there, the bottom left picture of Leon Goldstein, who was here for many decades, studying red blood cells. And what's interesting about non-mammalian vertebrates is that they have a nucleus in the red blood cell, whereas the mammals are enucleated, they don't have it. So it suggests that there's more function for these red blood cells in a non-mammalian vertebrate, and this is really intriguing. It's more of a comment than a question, I guess, but it's really intriguing that producing cilia may be part of that nuclear function that no one has really ever understood fully.
Yeah, definitely. Something we're not really sure what or why they produce cilia, but we think we have two ideas. Maybe they are circulating blood that get caught by the clot, by their cilia, or maybe once they get in the clot, they produce cilia at that time to help with their regeneration process.
Thank you.